Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hello, and welcome to the last podcast episode of the year. This week, we spoke to Robin Franklin again about stem cells located in our brains. The conversation centered around how these cells behave. Can they produce cells found outside the brain, which protect it from autoimmune attack? How does the brain respond to damage? How can a decline in cognitive function, which can be caused by aging, be slowed down? Without further ado, I give you part 2 with Robin Franklin. So why don't we just dive? Thank you for joining us today, Professor Franklin.
My pleasure.
I really enjoyed our last conversation, so
likewise,
looking forward to diving into some of the details of what we spoke about.
Yeah.
I'd like to start by talking about the progenitor cells we discussed last time. So one thing we spoke about was how these progenitor cells, their differentiation into these important oligodendrocytes, it gets worse with age.
Yeah.
But exactly how that happens, I'm not sure. So would you mind talking a bit about
Well,
that?
yeah. Well, nor am I, actually. I mean, we've described it as a phenomenon. What's very clear is that there won't be a single mechanism, a single explanation for why they differentiate less effectively as you get older. What we know is that the aging progenitor cell acquires all the hallmarks of adult stem cell aging. So, the whole physiology of the cell becomes dysregulated in some form or other. So energy metabolism is changed, DNA repair mechanisms are changed, senescence markers go up, and so on and so forth. And there are almost certainly epigenetic changes taking place associated with aging, which we haven't really explored yet, although I think there are groups elsewhere who have been looking into this in much more detail than we have. I mean, it's bound to be the case. I think one needs to think of these changes that are occurring with aging in a holistic way rather than a single mechanism way.
That's interesting. So what you're saying is essentially the mechanisms by which these aging progenitor cells lose their ability to differentiate are sort of the same mechanisms which affect all kinds of stem cells throughout the body.
I think so, yes. I mean, if I can use an analogy, I tend to think in analogies, and I don't know whether I used this one when we spoke last time or not, but if you think of a healthy function within a cell as being like a large symphony orchestra, and when it starts, everybody's fresh and all the instruments are in tune and the conductor's up for it and everybody's following the conductor and everybody has a bit of energy, and so it all sounds exactly as the composer meant it to, as a sort of beautiful, melodic, harmonious whole. And then as the orchestra carries on playing, as the hours go by, everybody gets a bit tired, the instruments go out of tune, and so the whole thing just becomes less and less harmonious. And I sort of think of that as a visual imagery of how cells are changing with aging. It's sort of it's not a single event that takes place, and it's not necessarily a quantum change that takes place. It's not a catastrophic event. It's just a general dysregulation. The whole thing just gets knocked out of kilter. It works with beautiful synchrony and when you're young, and then as you age, it all becomes it all falls apart, so to speak.
Going back to what you said a bit about how the aging process takes place. I mean, this isn't directly what your lab does, but it's related. So in your opinion, it's best to look at aging as a process which happens as a result of lots of different factors occurring in cells.
Yes, absolutely.
So if someone were to say it is sort of these epigenetic changes which are entirely driving the other hallmarks of aging, that might be...
Well, and of course, the epigenetic changes will be changing the transcriptional profile of the cell, and that'll be changing the protein composition of the cell, and that'll be changing its function. So, at some point, epigenetics are involved, but it's a real chicken and egg thing. So what's driving the epigenetic changes and generation? So it's probably the case that in young adult progenitors, the epigenome across the population is pretty similar, I would think. But then as those cells age and undergo a degree of replication, there will be an increased diversity in the epigenome, which is called epigenetic drift. So does that lead in adulthood to an increase in the heterogeneity of the progenitor population, such that some cells continue to regenerate very effectively, and some, as a result of epigenetic drift, lose the ability to regenerate effectively? Or does it actually lead to a decrease in heterogeneity? So now you have different clones with different selection criteria. And so it may be that in aging, what you do is you select out very specific clones, so that you decrease the diversity rather than increase the diversity. And there's some evidence in the hematopoietic stem cell system that this is what occurs with aging.
These are the stem cells in the blood.
Exactly, yes, yes. The stem cells in your bone marrow that make the cellular components of the blood. Exactly. It'd be very interesting to find out which of those two alternatives occur in brain progenitor cells. And of course, if the second model is correct, that there is selection of specific clones with distinctive epigenetic profiles, it may be that the criteria for selection of those clones is such that you're selecting for cells that have a decreased regenerative capacity, and so you're actually with age acquiring a population that are less effective at regenerating. But
these cells perhaps are better adapted to surviving as individual cells.
Exactly. For example, yeah.
I mean, do you see? So far we've spoken about progenitor cells as a catch-all.
Yeah.
Do you see different populations of progenitor
cells? So this is a really exciting question.
Developmentally, perhaps.
Yes. So the assumption has always been that the progenitor, a reasonable assumption, the progenitors are a single population of cells that all behave the same way. So we discovered a few years back that in fact the developmental origin of the progenitor cells reflects how they behave in the context of regeneration. So very simply, throughout the neuraxis, brain and spinal cord, the oligodendrocyte lineage has a dorsal developmental origin and a ventral developmental origin. It's two separate populations with two separate transcriptional machineries that make oligodendrocytes either in the predominantly ventral bottom parts of the brain and spinal cord versus the dorsal top parts of the brain and spinal cord.
So the best way to visualize that would be sort of the bottom of the nervous system being in the spinal cord ventral.
Oh no. Well, let's think of just the spinal cord, because that's where we did our experiment. So the bottom of the spinal cord, the bit that faces the viscera, if you like. Is ventral, and the dorsal, the top bit, is the bit that faces is closer to the skin and the back. Exactly. Yes. Yeah. So that's what I mean by top and bottom, dorsal and ventral. So a few years back, we took advantage of a really beautiful reporter mouse that had been developed by Bill Richardson at University College in London, where he used these different transcriptional machineries that drive the development of oligodendrocytes in dorsal and ventral to produce a dual reporter mouse. So where the ventral cells are green and the dorsal cells are red. It's a beautiful mouse.
So these are cells which have been engineered to fluoresce.
Exactly. Yes.
Due to sort of different proteins within them.
Exactly. So exactly so, Stan. They're engineered so they express a reporter protein that fluoresces a different color. So when you look at the spinal cord of these adult mice, the top parts of the spinal cord are red because they're predominantly myelinated by dorsally derived cells, and the bottom part is green, predominantly green because it's myelinated by ventral cells, although there is some mixing. So in predominantly green areas there are a few red cells and vice versa. So what we did was to say, well, if you demyelinate the adult and it repopulates during remyelination, do you see the same proportion of red and green cells when it regenerates, which would suggest that the cells have the same regenerative capacity? So we took the ventral white matter of the adult spinal cord, where the majority of the cells were ventral, and there's just a smattering of red cells of dorsal origin, and induced demyelination, let the oligodendrocytes get regenerated by the progenitor population. And what we found actually is in young adults, the dorsal cells respond much more vigorously than the ventral cells and make a disproportionately large contribution to the regeneration. So when that area of demyelination is now regenerated, there are more dorsal cells and fewer ventral cells than you would predict given the distribution before you induced injury. So that tells you that the dorsal cells have a different regenerative capacity than the ventral cells. And so the developmental origin is in some way predicting how they'll behave in the context of regeneration in the adult, which is very interesting.
And if we can better understand perhaps why one group of cells regenerates better, perhaps that will have implications for
exactly. Now, what's really interesting about that is that the implication of this is that the dorsal cells can spring into cell cycle and proliferate more extensively. And when we did the experiment again in aged animals, what we found was that advantage of the dorsal cells had been lost. So now in aged animals, the dorsal cells are responding less well than the ventral cells.
Less well.
Less well. So that's really interesting. We don't know why that should be the case. It's intriguing to hypothesize that maybe because they proliferate more, they age quicker, and so in the age system they've acquired more of the hallmarks of aging the ventral cells. Now that's just a hypothesis at present, but that would be an interesting thing to.
Now, can progenitor cells only form oligodendrocytes?
Well, if you put a progenitor cell in a tissue culture dish, you can get it to make pretty much what you like, and the only thing that you're restricted by is your own imagination. And in tissue culture, you can make a progenitor cell into a neuron. Now, the evidence that actually occurs in development or in any in vivo context is in the body, is pretty negligible. So, to all intents and purposes, these cells do not make neurons. Now, whether they make the other major cell type of the central nervous system, there are differences of view, different views and different data. Our experience in the rodent is that these cells can make astrocytes, but they do it very rarely. So they will make oligodendrocytes relatively easily, astrocytes pretty rarely. The human equivalent does seem to be more adept at making astrocytes than the rodent cell. Now, whether that's an intrinsic difference or because the context in which the experiments are done is not clear at present. Now, the other really intriguing thing we discovered a few years ago is that, in fact, these CNS progenitor cells are able to make cells of neural crest origin. So just to give you some context here, when you demyelinate the central nervous system, for the most part, the remyelination is by oligodendrocytes, as you would expect. That's the myelinating cell of the central nervous system. But in some clinical contexts and in some experimental models, you can get remyelination by Schwann cells, that are the cells that myelate in the peripheral nervous system.
Outside of the spinal cord in the brain?
Exactly, yes. So the nervous system is divided into a central nervous system, the brain and the spinal cord, and a peripheral nervous system, which are the nerves that go from it and to it.
The rest of your body.
Yeah, and it has a completely different developmental origin to the CNS. The PNS and the CNS separate from one another right at the beginning of developmental origin of the nervous system. So for the developmental biology aficionados, when the neuroepithelium is being formed and the neural tube, which is right at the point of gastrulation, it's very early in development, there are little clusters of cells which bud off from that, which are called neural crest cells, that then become the peripheral nervous system. So in developmental terms, as soon as you start to make a nervous system, there are two separate pathways. So seeing these Schwann cells in the central nervous system is sort of intriguing. It was first described, I guess, about 40 years ago when people started using electron microscopy to look at injury models in the central nervous system. And at the EM level, it's chalk and cheese distinguishing oligodendrocyte from a Schwann cell. It's very difficult. It's very easy to distinguish them. So the view is always that these were Schwann cells that had immigrated into the central nervous system from the periphery. So you bang a hole in the border between the CNS and the PNS, and the Schwann cells can then flood in. They're very migratory, very proliferative cells, and what's interesting is that they come into areas of the central nervous system where you've lost astrocytes. So astrocytes sort of defining whether you're in a PNS or a CNS environment. And if you don't have any astrocytes, and the Schwann cells will just make it a PNS environment. So that was the explanation for Schwann cells for many years. But then a while back when we were doing some experiments, again in collaboration with Bill Richardson's lab at UCL, using a reporter mouse, where we were really wanting to show that the progenitors were giving rise to oligodendrocytes, we discovered actually that all of the Schwann cells in the lesion were also expressing the marker which would suggest they're coming from CNS progenitor cells and not from the periphery. And to cut a long story short, we used a whole variety of different experimental approaches to demonstrate that that was unambiguously the case. Many of these Schwann cells that you see in the central nervous system are not coming from the periphery. Some of them are, but many of them aren't. They're actually coming from CNS progenitor cells. So the CNS progenitor cell, what seems to happen is that the CNS progenitor cell, when it pitches up in an area where there's an exposed axon and there are astrocytes, imagines to itself, in as much as a cell can imagine to itself, but imagines to itself in very anthropomorphic terms that it's in the CNS, so it makes an oligodendrocyte. But it sees an axon and there aren't any astrocytes, it thinks, blimey, I must be in the peripheral nervous system, so it makes a Schwann cell. So there's this really intriguing ability of a CNS progenitor cell to make a Schwann cell, even though those cells have separated so early on in development. So this is, to me, really intriguing, that a CNS progenitor cell will make a Schwann. And you might conceive of circumstances where actually making the remyelination peripheral rather than central would be advantageous, because MS is a disease that attacks CNS myelin and not PNS myelin. So if you made a patch of injury myelinated by PNS, it would be protected from the disease process. So there might be reasons, actually, to switch the progenitor cell into making a Schwann cell. And so that's something we're working on quite intensely at the moment, is to figure out what circumstances enable a progenitor cell make a Schwann cell. So and in fact, whether there's, going back to our earlier discussion, whether there's a subpopulation of progenitor cells that are more adept at making Schwann cells than others.
That's incredible. If you can make stem cells in the brain differentiate into peripheral cells,
yeah,
perhaps you can protect the brain from its own autoinflammatory disease.
Exactly so, yeah.
Once there's been damage in the brain, there needs to be a process of clearing that debris, the damaged sort of cell components which are still left. Is that an important process in allowing progenitor cells to form these oligodendrocytes and then repair the damage? Yeah,
no, it's really important. So any damage in any tissue in the body will trigger the innate immune response. The innate immune response to injury is primarily there to help patch things up. So remyelination is no different. When myelin is damaged, the innate immune response, in the first instance, the microglial cells, which are the cells of the innate immune system in the CNS, respond. And then if there's disruption of the blood-brain barrier, then you'll get recruitment of monocytes and monocyte-derived macrophages. So you will get a macrophage response to myelin damage. We showed a number of years ago that macrophage response is really important for triggering the regenerative process. And that at the time was regarded as a heresy in the MS field, because the MS field had been so hung up on the immune system being the bad guys. The notion there was a component of inflammation which is actually good for recovery seemed to puzzle people. Quite why, I don't know, because of course in standard pathology texts it's been written for years that the function of the innate immune response is to prepare the way for regeneration.
And these macrophages are literally eating up pieces of debris.
Yes. So they can be doing that in a good and a bad way. They can be contributing to the stripping and loss of myelin from a nerve fibre if instructed to do so by a maladaptive immune response. But they are also there to clear up the debris. Now, we showed a number of years ago that the myelin debris contains, this is work that Mark Cotter did when he was in my lab, the myelin debris contains inhibitors of progenitor cell differentiation. So as long as the myelin debris is hanging around, the progenitor cell's in an environment where they will struggle to differentiate because they'll be inhibited from doing so. So the macrophages come in and clearing away the myelin debris is releasing the progenitors from that differentiation block. So it's a very important part of the regenerative process, to say nothing of all the molecules that are produced by reactive microglial cells that are actually galvanizing the progenitor cell into an activated state where it can contribute to regeneration. So the innate immune response really as in all regenerative processes, I mean, this is not something peculiar to myelin regeneration, is fundamentally important to kicking the whole process into action.
So we've spoken about how the immune system can be acting as a good guy, essentially, or a bad guy in the brain when it comes to regeneration or damaging the brain.
Yeah.
Could you talk at all about how you could nudge? Is there potential to nudge the immune system one way instead of the other into a more pro-repair mechanism?
The innate immune system does what it's told to do. So in autoimmunity, in a context where there is an adaptive immune response, the innate immune response will contribute to whatever the adaptive immune response is. So I sort of take the view that actually one of the ways you can nudge the innate immune system towards being helpful rather than unhelpful is by dealing with the maladaptive immune response, which has been the strategy of drug development in MS and the successes in MS therapy over the last 20 years or so. So that alone will probably be enough to shift the innate immune response towards a helpful immune response.
You asked me to come back to how that changes during aging.
Yeah.
So let's get back to that.
So I described this process by which the macrophages clear away the myelin debris and so create an environment that allows regeneration to take place. What happens is, unsurprisingly, the macrophages become less good at doing what they're supposed to do as you age. So with aging, the macrophages become less and less good at clearing away the myelin debris. So if you look at injuries in old animals, there's still an awful lot of myelin debris hanging around. And we've shown that if you take the macrophages or the microglia out of the aged brain and give them some myelin debris to gobble up in a tissue culture dish, the older they are, the less effectively they do it. So the age effects on remyelination are not simply the age effects on the progenitor cell, but they are unquestionably also the age effects on the innate immune cell, and probably the innate age effects on other cellular constituents of the remyelined environment, which hasn't really been pursued as much yet. But there's now a lot of evidence that the innate immune response changes with aging in a disadvantageous way, and there is also some hints about how you can change that, how you can perk up the macrophage response with aging.
You can probably tell by now that I'm a little bit obsessed with metformin and the effects on how cognitive function change with age.
Yeah.
Do you think that it's possible within a given time frame that this will start to be something which is prescribed to people as they age, just as something to slow? Is that something people are interested in?
Very interested in, absolutely.
Their clinical trials?
Well, Stan, most scientists are extremely cautious, or should be extremely cautious about making these sort of claims, and certainly should be hesitant about making claims which really only clinicians can make. But I think this is which is
why I'm asking you to make one on the radio.
Right. So, okay. So my view on this is that we're on the cusp of a very exciting phase of this whole process of making new oligodendrocytes in the focus, and certainly my focus over the last few years has been in the context of focal myelin loss in diseases such as MS and how you regenerate those. But I suspect, the next 5 to 10 years, what will really emerge as interesting is how the de novo generation of new oligodendrocytes in the adult brain contributes to brain function and the acquisition of new knowledge, new motor skills, and so forth, and then how that changes with aging. And so I think a lot of the biology that we've been working on about aging and remyelination will have some relevance and possibly even direct application to what I suspect is going to emerge as an important area of myelin biology.
Because as we said last time, everyone has a vested interest in slowing age-related decline cognitively, and this could be a big mechanism for how that.
Yeah, I think it's going to be I think it's going to be an important element in an emerging story.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.